Hi, I'm Kathryn Jewett, and this is my Drosophila lab. I'm originally from Seattle, Washington, and I got my undergraduate degree in neuroscience at the Washington State University in Pullman, Washington. And that was a really great experience. I'm a first-gen college student. My parents did not go to college. And so I was the first in my family to go to college, and so it was a very new experience just trying to navigate how to get the classes that I needed, and I had no idea where I was going to go with this degree that I was really passionate about. But fortunately, I had the opportunity to work in many great professors' laboratories doing research with human subjects in psychology, as well as animal subjects in neuroscience, and studying sleep, which kind of ties into what I want to do now. But I really was grateful for those mentors that helped me, because I wouldn't have known where I wanted to go or what I wanted to do without their guidance. So it was just a really great opportunity to work in their labs. In the summer of 2020, I came to Juniata from the lab of Dr. Leo Palenque at the University of Washington, where I was completing my second postdoc. And it was there I first started working with Drosophila melanogaster, or fruit flies. So they're not just this pesky little bug that flies around your bananas, they're actually really useful tools to study science. The entire genome of fruit flies has been figured out. We know where all the genes are, and we can select genes that we want to study and target really easy using some awesome tools. The gene that I chose to study is called glucose cerebrosidase, or GBA. And the reason why I was interested in studying this as a neuroscientist was because mutations in GBA in humans predispose them to Parkinson's disease. It just turns out that flies also have the GBA gene, and if you delete this gene from the fly genome, then they have all these abnormal characteristics that are really similar to human Parkinson's disease symptoms. So we have things like trouble flying, trouble climbing, memory issues, they have a shortened lifespan, and what was really interesting to me is they have this buildup of protein aggregation, which are abnormal clumps of proteins within cells that can disrupt their functioning and even lead to cellular death, which, as you can imagine, is a really bad thing when we're talking about brain cells. So it turns out that a couple other labs studying GBA found that these flies lacking GBA also have a sleep phenotype. And so what that means is that they don't sleep like healthy flies do. And that was really interesting as well, because with human Parkinson's disease, one of the earliest clinical symptoms that is often found after the Parkinson's diagnosis is that these patients had a sleep disorder previously. And so there is a very clear sleep component to human Parkinson's disease, and now it looks like in these flies as well. So I'm really excited to bring my expertise in the sleep world from my undergraduate research and graduate research into my lab now, and connect sleep and GBA and see what effects GBA might have on these sleep characteristics. Students in my lab will use biochemical assays like Western blots and PCR to assess how different cell types may be contributing to protein aggregation. They'll also learn how to isolate extracellular vesicles and identify their contents. Students will also perform behavioral assays to assess climbing ability and record sleep-wake cycles of the fly. I'm looking to be a mentor to my students just like I had, because without them, those individuals, I never would have gotten to where I am today.